Okay, so I recommend having this open in a view where you can take notes underneath, rather than having it on the slideshow version of the view. I would just slide it up so that you've got, there should be space for notes. And Nate's going to wave his laptop in the air to show you what you should be looking at.
That's not terrible, John.
You don't have to track me though.
He's not actually teaching yet.
Okay, listen up. So last elective, Jolio introduced you to hormones in terms of hormone signaling, and today basically what we're going to do is we're going to look at signaling within a cell. After this lesson, next Tuesday, he's going to be following up with more hormonal systems, more feedback loops, that kind of thing. But this one basically links into what I'm going to be teaching you, which is about genetic control mechanisms, what switches on and off genes, and how that works. Okay. In order to understand this, we need to be thinking, okay, how are messages being transferred? How are cells communicating with each other? And then, how is that message? Basically, we're doing kind of arrival at the cell. How is the message arriving at the cell? How's that all working out? Okay. So, cells send messages, okay, just like you do. And there's various ways they send messages. They can be messages that are sent in direct contact, short distance, or broadcast. So direct contact is the equivalent of passing a note to your neighbor. The cells have to be right next to each other, and it's physical interaction between the two cells. And we'll see examples of these in a moment. Short distance is the paper airplane equivalent. Okay? There is a gap, but it's very narrow, and this is still a specific message going from one cell to another. Okay, so I wanted to send a message to Bailey, I just throw a paper airplane across to her with the message on it. We don't have to be direct, physically next to each other to pass the message, but it is still a message going from one cell to another directly across a short gap. Okay, so the cells are pretty close together in this instance. And then lastly, we have a broadcast message. A broadcast message is the equivalent to what a hormone does. It sends this message out across your entire body saying, this needs to happen. Or I'm going to put this out there for the whole body to know. But just like if you post something on Facebook, okay, which can go out to all of your hundreds of friends, only a few are going to necessarily respond, okay? Maybe the few that message applies to. And in the same way, when you send a hormone out in your body, only the target cells are going to respond to that. And this is all to do with the proteins that are on the surface of those cells, the specific receptor proteins that are going to pick up that message and go, okay, that's relevant to me, I need to act on that. So this is not like sending a letter, okay? It's much more like doing a status update on Facebook, okay? It sounds silly, okay? It's much more like that because it's throwing it out there and saying the people who this applies to need to respond. The rest of you can read this and just go, okay, this does not apply to me. Okay, so these three types of message. So here we have the three. We've got the direct messages. This is the equivalent of being cell to cell, right next to each other. Two of these, paracrine and synaptic, these are both these short distances that are like a paper airplane equivalent. And then we have the endocrine, which is the hormonal systems. So essentially what's going to happen today is I'm going to talk about what happens when these messages get to the cells and how those cells respond to the messages. Jolio, in his elective presentations next week and the weeks following, will be looking at hormones and the nervous system. So he's effectively looking at how are these messages traveling through the body, where do they go between. I'm focusing in on the minutiae, the tiny details of what happens when these messages get to a cell. Because later on that's going to inform what switches genes on and off, what determines which genes are expressed in your body and which genes aren't expressed. Can I just check? We go to 12:30 today? What time do we go? We go to 12:30? Thank you. Okay, so direct contact. Both animal and plant cells have cell junctions between them. Okay? These are direct connections between the cytoplasm of adjacent cells. Okay? In some cases, the cytoplasm is actually able to flow from one cell, the liquid cytoplasm is able to flow from one cell to the neighboring cells. These are called gap junctions when it's in animal cells. In plant cells, these are called plasmodesmata. Okay. This is direct contact. That means that if a molecule is produced in one of the cells, these cell junctions, if a molecule is produced in one of the cells, it doesn't even have to go through a cell membrane to get to the other cell. Okay. It doesn't have to pass through any kind of cell membrane. It can literally just flow from one cell to the next. The cell membranes of these cells are interconnected. Which starts to kind of slightly, possibly blur our definition of a cell. Because we've always thought of cells as being whatever, the membrane. If you want to take this to a real extreme, you'll start to look at fungi, which are polynucleated cells. These are huge cells that have lots of nuclei in them. And it's like, well, is that one cell or lots? Scientists generally would say that's one cell with multiple nuclei in it. But it's like taking these gap junctions and plasmodesmata to an extreme, where basically the cytoplasm can just flow up and down this really long cell. Okay. So this is direct contact, and of course it's pretty quick. Another good example of this is antigen presentation. While Jodie is teaching about hormones and stuff, I'm going to be doing stuff on DNA. After I've done stuff on DNA, I'm going to be teaching about the immune system and how the immune system works. We have one of the coolest science videos ever to show you. We'll also be acting out the immune system, and you'll get to become one of these cartoon characters, not necessarily one of the guys here. But this is just a nice example of some direct communication between cells, which we will return to when we cover that. This is a phagocyte. Okay, remember phagocytosis means what? Phagocytosis? Eating, yes, cell eating is taking in a solid. So a phagocyte, this is a type of white blood cell, dendritic white blood cell, takes in, it consumes a bacteria. What organelles would bind to that vesicle that contains the bacteria now? It absorbs the bacteria in a vesicle. What kind of organelles would bind to it? Solvents. Lysosomes. Why?
Lysosomes digest material.
Yes, what kind of enzymes do we say they contain? Yeah. Hydrolytic. Remember hydrolysis? It's the breaking down by adding water. Hydrolytic enzymes are going to do that. So they destroy the bacteria. But they don't destroy it completely. It's pretty cool. They chop it up into lots of pieces. And then they transfer these pieces to their outer cell membrane and they hold it out like trophies. Look what I did. Look what I chopped up. Okay. This is important in your immune response. Because these fragments of the bacteria that have been left over, being held out, are spotted by these cells called helper T cells. Helper T cells are a different kind of white blood cell. Helper T cells come along and they make this direct contact and they're like, okay, it's like coming over and feeling it and going, okay, I see what you did there. Okay, you're saying this is dangerous and we need to destroy this? Right, I've got that. And it passes that message on. Okay, it's going to spread through, it becomes what we call described as being activated, and that's going to have knock-on effects later on in the immune system. But I'm not going to go into that now, we'll go into that when we get there. The key point here is this is direct, it's a nice example of direct communication. One cell interacting immediately with the other, holding out this broken fragment of the bacteria and saying, look at this, look what I've got. Okay, you need to find this and destroy it because this thing is attacking our cells, this thing is attacking our body. Yes?
Does that helper T then send out a broadcast message to the other organ?
And SOS to the world, something like that. No, that's perfect. Yeah. Okay. So, short distances. That was direct contact. Now we're going to start talking about short distances. There's a couple of different examples of this. Paracrine signaling is not something I've come across a lot. It says local regulation. So this is kind of like this group of tissues needs to follow these instructions. So I'll send it out and this is so there's tissues around me saying, hey guys, we need to grow, or we need to do this. It's not long distance, it's a local communication mechanism. The other classic example, which you're going to do with G in detail, is signaling between nerve cells. Nerve cells are not in direct contact with each other. There is a tiny gap called a synapse between the two nerve cells. And neurotransmitters are these chemicals that are released, acetyl-CoA, nice example of that, is released from one out into this gap, across the gap, now it's a very, very tiny gap, to the next nerve cell. That's happening in your brains right now, all over the place, really fast. These little connections between the nerve cells. And it can actually carry out some really cool kind of computational biology, because what we have is, you can have one of these nerve cells saying, go, and you can have another one here producing an inhibitor that says, no, don't go. Okay, and so we can start to make decisions on the very smallest scale. It's like, okay, if the one who's firing is saying go, and the other one isn't firing, then I'll go. But if this one's firing and this one's firing, then I'm not going to go. And if this one's saying no, you can start, it becomes a decision matrix, if you like. And in that decision matrix is where your conscious thought exists. It's where your decision making, both on a very basic level, like, ooh, this is a hot pipe, I'm going to move my hand away. I don't have to consciously think about it, I automatically do it, or I jump at a loud noise, through to the very complex decisions of conscious thought that we make.
Is there a reason why they're not in contact with each other?
Absolutely. What does this enable? Having a gap, what does it enable? Yes? The messages to be blocked and controlled. It's a control. Interestingly, and I've asked you to cover this as well, various chemicals, drugs that you take will impact this interaction here. Alcohol has an impact on this.
This is an inhibitory GABA.
Yeah, if you can inhibit and slow down nervous function, which is why people who have bad coordination if they're drunk and they make poor decisions. So different drugs, and that's not just drugs that you consume and take into your body, but also drugs that your body produces, such as adrenaline, can affect the way that these work. So these are examples. As I say, the examples you're going to get to, you're going to get are going to be much more specific. But and then lastly, we have these broadcast messages, these hormones. They're sent from one cell or tissue out into the body, and that message is going to spread right through the body, and whichever cells have the correct receptors are therefore then going to respond. Okay, so cell signaling. We're going to focus on what happens when the messages get to the cells. And there are 3 stages. Another guy winning a Nobel Prize discovered how the hormone epinephrine acts on cells. Does anyone know what the other name for epinephrine is? Yes? It is. It's adrenaline. Good knowledge. Okay. Epinephrine is the same as adrenaline, so he won the Nobel Prize. He stipulated that there are 3, Sutherland suggests there are 3 processes that are involved in cell signaling. We have our initial reception, our transduction, which is like the processing of the message, like what happens to it when it gets to the cell, and then the response, how does the cell react? And there's lots of different ways a cell can react. It can send a signal on, it can produce a particular protein, it can release a digestive enzyme, it can do all kinds of different things. Okay. So here's a nice overview. We have reception at the start, so this is the extracellular fluid, this is outside of the cell. Here's our receptor. It's going to take the signal molecule, which is going to be specific, causing reception. Then we're going to get a series of chemical reactions in a pathway, called the transduction pathway, until we get our response. Okay, what is the cell going to do? So step 1, reception. This normally causes a conformational change in the protein. It's going to bind to a protein in the cell membrane. Remember, the cell membrane is covered in lots of different proteins in the fluid mosaic. And it's going to bind to a specific receptor. One of the beautiful things about proteins, of course, is that we can have
billions of different types of molecules. It's just endless possibilities. Almost, you can think of it as virtually infinite, the number of different shapes you can produce with a protein. And those of you who've been playing that game know that there's a lot of ways you can mess up. That protein has a very specific shape that will bind to the specific shape of the receptor. What does this remind you of? What does it remind you of? James? It's very like enzymes and substrates, except this signal molecule is not going to be reacted in some way. Okay. Instead, do you remember the induced fit model? Do you remember when the substrate binds to the protein, it causes a change in shape of the actual protein? That's exactly the same thing that's going on here, except that's not causing a catalysis or anabolic reaction in the signal protein. Instead, the change in shape of the receptor is going to send a message into the cell. This is a very highly specific signal, causes conformational change, as I mentioned. And the receptor protein can either be in the membrane or it can be embedded actually deep inside the cell. Kind of strange. The actual receptor could be inside the cell, depending on the type of hormone. Transduction, we'll come back to that. The binding of the signal molecule alters the receptor protein in some way. We already said that. The signal starts a cascade of reactions. What does that mean? Cascade. Yes.
Like a series of lots of reactions taking place.
Yes, I would use the word chain if I was using that description. Though, why is cascade? Cascade is different. Yes.
Like one makes a bunch more, makes a bunch more, makes a bunch more.
Yes. Think about this. If you've got just a few cells in your body releasing a hormone, there's not a lot of molecules being produced. So when those hormones get to your target cells, it's going to be a tiny little message. And if it was like a chain where that one molecule made that one enzyme work and that one enzyme did that thing, it's going to be pretty slow. You're not going to see a big, dramatic, quick response. The kind of response that you need if a bear walks through the door and you need adrenaline to go through your body and tell your body to respond, run, fight, whatever. Okay, fight. You have chosen poor. You know, they did, there was a while in the US when they tried to have different fights between different animals just for fun. To see, you know, it's like what happens if you put a tiger into the bear or a lion with a gorilla? Like, what happens in those different situations? And there is one hard, fast rule. The bear wins every time. It will kill whatever you put in there. It doesn't matter if it's a Bengal tiger, it doesn't matter if it's a lion or a crocodile, the bear wins. Bears are ridiculously tough. You do not want to do it. On the other hand, an easy video to watch, bear fighting. It exists. They declaw the bears, they don't let them bite anything, they have them muzzled, and people wrestle bears. There was actually a bear wrestler who fought actual wrestlers. Because they removed the claws, it was pretty cruel.
But can you still just
So occasionally they would kill people, yeah. The bear only won. There's one famous bear that did. The bear only lost one fight, and when it was little, against this massive guy. But generally they won. So this is all about, anyway, back to this. This is all about signal amplification. This is all about taking that tiny message and going, this is important, the cell needs to respond, and it needs to respond now, it needs to respond dramatically, as opposed to that tiny little message that came in. So step 3, response. Cell signaling leads to regulation of cytoplasmic activities or transcription. Transcription is what? Come on, what is transcription? Devin? RNA synthesis, yes, making RNA, okay, which is going to make a protein, which is going to create enzymes or different proteins that we need. Okay. Or it could just be controlling something that's happening in the cytoplasm. Often, in a lot of cases, there's already something ready and waiting in the cytoplasm for that particular response. That's the case with your nerve cells and other cells as well. There's a little thing in the cytoplasm ready, waiting to go. You don't have to bother with transcription. It's just there, sitting in a vesicle, ready to be released onto the world. Okay. Cell signaling pathways create a variety of cellular activities, there's lots of different ones. So I mentioned that some receptors are found inside the cell. Giorgio, in his lecture last time, he mentioned that you have not only protein, hydrophilic, most protein hydrophilic hormones, such as adrenaline. You also have hormones such as testosterone, which are fat soluble. They are non-polar. How does that mean that things are going to be different when it comes to that arriving in the cell, Henry?
I'm going to be able to cross through the cell membrane.
It will. It's going to be able to fit between the fatty acid, phospholipid violet. Okay, those tails, because it's non-polar. And so the receptor will be inside a cell rather than on the surface. Okay, so here's an example of one coming in, binding to a particular protein, and that protein then moves off. We have the inhibitor will drop off it. That causes transcription. Okay, we call these transcription factors. So here's our example of testosterone. Comes into the receptor protein. It's able to pass through the plasma membrane. It binds to the receptor protein in cytoplasm, activating it. This goes into the genes. It's called a transcription factor, and it causes these genes to switch on. Those genes are then going to produce the mRNA, which are going to produce the protein, specific protein that might be to do with production of hair or deepening the voice or whatever protein is associated. Estrogen is another one that is also fat soluble. These steroid hormones, you may remember that steroids belong to the lipid. Okay, cell surface receptors. So we get lots of different types. These are the types I'm going to talk about in a moment. So let's just move on. Transcription factors, switch genes on and off. Okay, so sometimes it'll go straight in and just copy the gene, and that's what it does. Other times it'll switch on transcription factors, which then in turn produce a chain reaction of producing, transcribing the gene over and over and over again. Termination of the signal. So you don't want the signal to remain forever. Okay, if you send a hormone out, you don't want it to keep going on and on forever. Otherwise, you'll be teenagers for the rest of your life. Something like that. So basically, what happens is after a while, it's going to drop off the binding is going to reverse, and it's going to drop off the receptor. So the three types of membrane receptors we're going to talk about: G-linked receptors, ion channels, and tyrosine kinases. In terms of order of likelihood that they're going to come up in the AP, it's pretty much that order. I would say G proteins and ion channels are much more likely than tyrosine kinases, but anything could come up. So G proteins. G proteins are relatively straightforward. There's a number of different steps that take place in the G protein. We have, here's our G protein linked receptor. There's the G protein. It's holding onto a GDP. Hang on a second. GDP. This is not gross domestic product. Okay, GDP. What do you think it stands for? Is there anything we've heard of before that sounds a little bit like a GDP? Perhaps. Sure.
Guanine diphosphate?
Yeah, it's something like that. I can't remember exactly how they phrase it, but it's the equivalent of ATP, but with G instead of A. It's a diphosphate. So what's its job? What do you think it's going to do? Yes?
Energy carrier.
Yes, it's going to be an energy carrier, but interestingly as well, it's also going to act as a signal. Okay. Like you could use a flash of light as a signal. This one can often use energy changes as a signal too. So what we have here are proteins. So what happens? First up, the signal molecule binds to the activated receptor protein. This causes the GDP to be rejected, and a GTP binds on. TP stands for? Triphosphate, exactly, okay. This change causes the G protein to detach from the activated receptor and kind of start gliding across the membrane. It binds to another protein, and that's where the fun starts. Okay. It can bind to an enzyme, or it can bind to an ion channel. It can bind to different things, and the response that comes from that will be different. Once it is bound to it, it's going to release the phosphate, and the GDP is going to go back to the receptor. Notice the receptor has lost its signal molecule, its hormone, and so the GDP will come back to it. Okay. Question?
So this, in short, the G protein helps control how many phosphorus ions are in the cell?
No, that's not its purpose. The G protein, this is just a little signal that comes across, and the signal provides, this is actually providing the activation of the enzyme with a little bit of energy in order to change its shape.
So this is just how an enzyme can get energy to do something. One way it can do that.
Yeah, it's going to cause a shape change, it's going to activate the enzyme effectively.
Alright.
So, here's a slightly different example, a nice video showing a slightly different example. The video is linked on the PowerPoint right here, but I'll show it to you this way. So just hold on a second. Now, the reason this is going to be a little bit tricky for you, but this thing here, these are the three parts of the G protein. This is a tiny bit more complex than the way I was explaining. You don't need to know the level of the fact that there's three different parts and one splits off, just as it is this one.
The receptor site on the outside of the cell membrane, the G protein changes conformation, and guanosine triphosphate places the guanosine diphosphate on the alpha subunit of the G protein. The activated alpha subunit then separates from the beta and gamma subunits. The alpha subunit, with guanosine triphosphate attached, binds to the calcium ion channel, causing the calcium ion channel to open. Calcium ions diffuse into the cell and combine with calmodulin. The combination of calmodulin and calcium produces the response of the cell to the ligand. Phosphorylase
So you just notice here, this is not this time activating an enzyme. Instead, it's doing something different. It's opening a gate, allowing ions through. This is a common response.
activity removes a phosphate from the guanosine triphosphate, leaving guanosine diphosphate bound to the alpha subunit. The inactivated alpha subunit separates from the calcium ion channel, and the channel closes. The alpha subunit recombines with the gamma and beta subunits, and the G protein recombines with the receptor.
How's this an example of a cascading effect? How's this an example of a cascade? Or just see? Yeah.
Each part of the process is effect of transport.
That's more like a chain. That sounds like a chain to me. How is it more like a cascade? Yeah.
Because the one leads to opening the channel, which leads to all of the different reactions. So one.
Or in that particular case, like I'm focusing on this particular case. Why we started off with one molecule. Alana?
Well, there was one molecule that attached to the, I don't remember what the receptor was.
Just the receptor. That's fine.
Okay, and then lots of other ions came in, even though they weren't being.
Okay, so the ion channel opened up and suddenly lots came through. So there's just one molecule coming through. So it's just being like a chain. We're getting a cascading effect happening. Okay. So this is just a diagram showing effectively the same thing. So these types of receptor proteins are literally just taking the thing and then shifting it. Okay, so this is just more detail on the same thing. Protein kinases. So a nice example using protein kinases is the one that we covered in block period, insulin. Insulin is picked up by protein kinases. And these proteins in the membrane, they come together, they give each other a high five, causing phosphorylation of these proteins. That causes glycogen synthase to work. And glycogen synthase does what it says on the tin. It makes glycogen out of glucose. Yes?
When you said they get phosphorylated, does that mean they give off phosphate groups for everything else?
They phosphorylate the other. So
Oh.
tyrosine kinases give other things to the phosphate group saying react. Okay, and then they start to change. Yes.
A quick question. So this is a kinase reaction,
Yeah.
a kinase receptor reaction,
Tyrosine kinase.
tyrosine kinase, and this. Sorry, my camera's off, but then that's when two separate what is it? Two separate
Protein structures
come together, and then that activates them. Yes.
Okay. So this is to show basically that we have a chain reaction that follows. Often it's not just going to be one molecule produced and then you get a reaction, but one molecule will be produced and that will enable another reaction. So you can see here, RAS, okay, enables RAF to happen, which enables MEK to happen, which enables ERK to happen, which causes these response proteins which go in. If we look at it this way, let's just skip through, this is the same kind of thing. What we're effectively getting is our signal and then this cascading effect coming down. I have another video here to show you how to show it. See if we can find that.
Receptor proteins interact with signal molecules at the surface of the cell. In most cases, the signals are relayed to the cytoplasm or the nucleus by second messengers, which influence the activity of one or more enzymes or genes inside the cell. However, most signaling molecules are found in such low concentrations that their effects in the cytoplasm would be minimal unless the signal were amplified. Therefore, most enzyme-linked and G-protein-linked receptors use a chain of other protein messengers to amplify the signal as it is being relayed. In the case of a protein kinase cascade, one cell surface receptor activates many G-protein molecules. Each G-protein activates many adenylyl cyclases. Each cyclic AMP in turn will activate protein kinases, which then activate several molecules of a specific enzyme. The end result is rapid production of high levels of the final product.
Okay, so something is mentioned there called CAMP, cyclic AMP. The C stands for cyclic. So often what will happen is when the GTP moves across, it causes ATP to be made into AMP. What do you think AMP is? Yeah.
Monophosphate?
Monophosphate, absolutely. What's the first word? Adenosine monophosphate. Okay, so like ATP, adenosine triphosphate. Here we have adenosine monophosphate. Okay, the ATP is broken down into that. AMP is used as one of these cascading signal molecules. Okay? And we get a lot of it in the cell. So we've got about 7 minutes left. I want to act this out. Acting out things is always fun. Okay, so what we need is somebody to be a receptor. Jack. Jack is a G-protein linked receptor. We may not get onto it, but can I have two people who would be kinase receptors? We've got a kinase receptor and a kinase receptor over here somewhere. Okay, we need someone to be an ion channel. There's an ion channel. We need someone to be an enzyme. And we need someone to be a G-protein. There's our enzyme. Okay. Listen up. Tony, you're going to be in charge of protein hormones. We're going to need some. Let's see. Can I put you in charge of ATPs? have some ATP. Lots of ATP. Okay, you be in charge of TP and GDP at the moment. Okay, and I need everybody who hasn't got one of these things to get some scratch paper out.
Can I turn on the light?
And I would like you to rip it in half. And then rip it in half again so you have 4 pieces. And screw it up into 4 little balls. Okay, you got that? Quick, quick, quick. That's 5 minutes. Next, I would like you to take all of your resources. And anyone with a sticker on their head or with some balls is going to stand on the cell membrane. Everybody else is going to sit on the floor on this side of the cell membrane and this side. Let's go. You guys just sit on the floor over here. That's fine. ATP, you could just sit right there. Just hold on to those there. That's fine. G-protein right here. G-protein link receptor, link arms right here. Okay, that's great. Kinase receptor, I'm not sure we're going to have time, so sit down. Kinase receptor, sit down. Enzyme, yeah, we'll have the enzyme right here. Okay, I need you to scoot back just a little, all of you. Okay, can I have someone who's going to be some transcription practice?
This so everybody knows what you're doing. What does yours say?
Okay, so this is essentially what's going to happen. You're probably going to want to turn the other way. That's it, link that way, that's perfect. Come across here a little bit further. Ion channel, actually I'm just going to get you to step out the way. Well, actually we'll do the ion channel first, enzyme out the way. Okay, so what happens with an ion channel response? Protein hormone goes to the receptor. Oh. That's GTP. Okay. You're going to switch out your GTP. Hang on. No. Track me. You're going to switch out your GDP with GTP. So just switch that to Jack. And Jack, away with you. So say G protein. Say G protein. Push her away. She's going to push all the way over to here, and you go, and you say, open sesame. Okay, the ion channel is going to open, and through all the ions come. Okay, that's our first response. Can we just do that again, one more time? Actually, really, you should say G protein and push her away just before she does the switch. So as you're moving across, right back. You ready? Protein hormone arrives. Okay? Got it? That's the ion channel. Right, enzyme. Ion channel, sit down. Enzyme, where are you? Come on in. Are you ready? Okay, I'm going to explain this one step by step, Chris. This is a similar kind of cell signaling we've got going on here. The hormone's going to reach the receptor. You're going to go G-protein. You're going to switch to TP just like you did before, except this time you're not going to say open sesame. You're going to say AMP, please. Okay? AMP. You want the cyclic AMP. She's going to get here and say AMP. At that point, sure. You're going to throw Myra a ball of ATP. Throw it down, Myra. Myra's going to rip off 2 phosphates, and she's going to throw it at one of you. Okay? When she throws it at you, you're going to increase the cell signal. Basically, if you're one of the first people she throws it at, I'd like you to throw your pieces of paper at somebody else around here. If you get hit by a piece of paper, you're going to turn around and throw it at the transcription factors. If you get hit by a piece of paper, you're going to start running up and down the wall because you are transcribing DNA like you got it? So quick recap. We're going to do a hormone. Just back and forth. We're going to get the hormone to hit the receptor. The receptor is going to release the G protein, which is going to switch G proteins over here. You're going to say 2. And you're going to keep passing the 2, okay? As soon as she's thrown one away, give her the next one. Okay? You're going to say make AMP. AMP is going to be the signal, so you're going to take AMP from him, rip off the 2, throw it at somebody, they're going to throw a piece of paper at someone else, they're going to throw their piece of paper at the transcription. transcription factors back. Once you've thrown one, take the next one. You can throw it at whomever. You got it?
I have two cameras working.
Okay. Let's see how this works. I want to hear the different parts. Go. Hormone. Sorry, start again. Okay, yeah, you need to be able to hear you. You shout protein hormone that comes in. Okay. Don't forget to really yell out your parts. Let's go again. Protein hormone. G-protein.
Make ADP.
Okay, stop. We reset very quickly. ATP's back over here. Let's all respect back quick. We're going to do this one more time. So a couple of notes. I think those are your paper. You need to remember, if you hit my ball, you're trying to see your paper. If you hit my paper, you turn around and throw those guys. Okay? If you shuffle back this, you're trying to throw. Just over it. Okay, a couple of notes. Chad. Could you shout GTP when you throw it? Yeah, she's got enough to say. And when you start throwing, you say AMP. No, you shout ATP. You're going to shout AMP when you throw it. Okay, you got it? So you're just saying ATP and you just say AMP.
I say make AMP.
You say make AMP and that's when you start throwing. Okay? Let's see if we can do this. Nice and loud. I want to hear every step. Go. Protein hormone.
G-protein.
Make AMP.
ATP.
AMP.
ATP.
AMP.
ATP.
AMP.
Come on, guys.
AMP.
Not good enough. I want to see it one more time. One more time. Reset. One more time. Reset, please.
Here you go, Sean.
Okay. This last time.
You're doing a great job.
This last time, if you get hit by a piece of paper, right in the middle, I want you to say transcription factors and start throwing at them and say, I'm transcribing. Okay? You guys have got to say transcription factors and they're going to say, I'm transcribing. You got it? We all good?
I'm gonna have to subtitle all of
We
these.
ready? I want to hear it nice and loud. Here we go. 3, 2, 1, begin. Protein hormone.
G-protein.
Make AMP.
ATP.
AMP.
ATP.
AMP. Oh, sorry.
Solomon, wake up. Let's go. So starting with one hormone, we had a cascade effect until there were loads of transcription factors and we're producing lots and lots of copies of those
Now
genes.
you guys transfer.
Hang on. Just one last thing. This is a lipid hormone. How would this be different if it was a lipid hormone? Yes. The receptor would be on the inside of the cell. We could skip out the G-protein start part. I'll give it straight to him and he can start throwing the AMPs out. Okay? Sorry. Can we get all the bits of paper up? It falls into the room. We're done. That's it for today. Good job.